We have observed, very strong oil control functions, and that's also, from deeply understanding DHT.
It should be 6 months study. Normally, we're talking about from beginning to end 1 year, recruiting those kind of things. And I can tell you, AGA patient is not difficult to recruit at all. The reason for the 2 years is because, first of all, as you probably know, the COVID situation in China, that have a huge impact. We were forced to stop the study, pause the study, let alone to say other factors. I'm just going to list a few.
Can you explain, just to the audience, to the uninformed, how does GT229 work? Like, what is this? KX826, it just stayed above baseline in your phase 3 1 year KX826 results. It did stay above baseline there.
I'm very interested in learning your background. The reason I'm asking the question you ask is really insightful and very good depth.
It was when I started dealing with the condition, androgenetic alopecia myself, I got back into looking at clinical trials and looking at the literature, and I kind of became obsessed over it after a while.
Sorry, I'm just carry over. I think that you're great.
Hello, everyone, and welcome back to another episode of the HairStacks podcast. And today is a very, very special interview. We have some representatives from Kintor. I'm sure many of you guys are very familiar with Kintor. Right now, they have KX826. I think it's released as Koshine 826 on their mall website. The link to that will be in the description. But they also have other drugs that are in development, primarily one that a lot of people seem to be interested in, not only just being KX826, but GT20029. So I'm now going to pass the floor on to the people that I'm interviewing. Now I'll ask if they can please introduce themselves to the audience. Before we start this video, please consider sharing this with a friend, liking the video, and of course, subscribing to the channel. And for additional channel support, become a member for as little as $2 a month. But seriously, try not to skip around too much in this video because you'll need the full context to understand everything. As always, check out the links in the description below.
Yeah, absolutely. Maybe I start. My name is Grace Wang. I'm actually based in US, and my background came from clinical experience and also cross academia fundamental research couple of years. So I've been working as clinician in internal medicine training, and I've been working as a fundamental research scientist. And for the clinical research aspect, I've been working in sites, clinical research sites as investigators and clinical monitorings, and also work in the different sides of CRO, which is clinical research organization and industry. And my experience has been back to 15 years. And I also have a PhD on molecular biology. And it's my pleasure here to be able to have the chance to communicate with everybody who is interested in the Kintor products. Yeah, I will pass to Wilson.
Hi, my name is Wilson Lou. I'm a physician by training. I graduated from Beijing Medical School and come to the United States and got my PhD with the University of Pennsylvania, and worked in the industry for more than 20 years. I worked primarily with a big pharma and then CRO. About 4 years ago, I went back to China and joined Kintor in about 2 years ago, working on this product for the past few years and also portfolios. I'm mainly for all medical aspect of the product. Glad to have a chance to discuss with all of you.
Yeah, thank you guys so much for introducing yourselves to the audience. Some really, really impressive backgrounds. I guess just to kind of ease into the interview, a question for both of you guys. How did you find yourself in the pharmacological sort of research field?
I can get started. I think that, once I finished my medical school and joined, thinking about what I'm going to do in the future, and think about all those things, one thing coming to my mind is that how to help people. That's my sort of reason for joining pharmaceutical company. If you look at the industry, there's of course different kind of things, but one thing stand out that can impact everybody's life is medicine as well as drug. We're all human. We all have a disease sometimes in our life. Treating those um, diseases is, I think, one of the most honorable profession in the world. So that's primarily the reason I joined the pharmaceutical industry. I think that's very 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 fulfillful.
Yeah, definitely. I find myself in the pharmacological research, pathway is, from my background, let's say, I've been trained as internal medicine, and also I've been years fundamental research. So I find myself, the clinical research is the best way for me to be able to get as close as to the patients, not only by the research purpose, but also from, clinical recognition. So let's say I hope, and I do found with recent 15 years as a medical scientist, I feel like my background from both settings can absolutely give a good combination in the pharmacological research, pathway, and in the drug development pathways. So not only from the disease, character itself, but also found how those interesting proteins or molecules, how those compounds be transferred to the actually research through the animal, through the cell, and eventually through the human being, and eventually get accessible for those patients. So that's my passion is, and I just found I am myself in the right place. Yeah.
Thank you for the responses. The next question would be more so about the discovery process. So, what is the drug discovery process generally like from a sort of high level kind of overview?
Well, drug development is very complicated process. It starts with a concept. Used to be you have to understand the pathway before you can sort of think about inhibitors, agonists, antagonists that can either help or block some receptors. That's old school fashion way. They do lots of screenings like in KX826 and try to find some hints, some clues as they call the concept. And then once they do, they did a lot of preclinical studies, animal studies, try to see whether we can demonstrate some efficacy or at least some signals in animals. That's the first step. Then the key step starts. Then they have to do in this part, in previous part, you can do it in any lab. Then the key step starts, and at which time you're going to spend lots of money. One of the key things you need to do is try to see any toxicities. There's a lot of labs can do that, but they call this GMP certified toxicology labs. So what they do is they're going to use a standard way to test any toxicities that you can see in the animal. So based on that data, you design studies that can start as a human. Once in human, then they start as phase one. Usually we're talking about a very small amount of patient. As you can see in our KX826, we also did this phase one study. We're talking about 20, 30 patients at different doses. Then move on to a phase two. In phase two, you're going to have a larger, normally 50 to 100 patient. The key of that is they understand whether in the ideal situation the drug can work. Once everything's settled, you move on phase three. Now in phase three, we're normally talking about hundreds, if not thousands of people. The question you want to answer in phase three studies are try to see whether in general population it can work. Obviously, when you go to a general population, there is a different kind of issues. Everybody is not the same. So once you pass that, normally you're going to have enough patients. Once you pass that, then you're talking about regulatory filing, going to the different countries for approval. Because Though we are all under the ICH, different country has different pathways for approval. You normally, this is a country by country process. You go to approve in one country, and does not guarantee you are going to be approved in another country. Then you go to market. Then go to market. There's also a different set of issues. In the clinical studies, which I'm going to mention later on when we discuss, we're talking about select group of patients. No matter it's 500 patients or 1,000, it's still select group. In the real world, then the people are different. People use your drug off label. People may not have the target disease to use it. They're going to have different issues. You're going to facing adverse event. It could be due to the drug. It could be just normal happening in life. So these are the things we have to deal with. So those are just very quick summary of the drug development process.
Thank you so much for giving that summary. That was very succinct. I guess my next question would be, what are some of the major issues or challenges when it comes to the delivery of the drug for either or both oral cases as well as transdermal cases?
There are, there's lots of different ways. You just mentioned two ways of delivering drug. There are different ways of delivering drug. Each has its advantage and a disadvantage. Oral, for example, we normally take pills, aspirin, whatever pills you take to control whatever you want to control. For example, hypertension. Those pills, when go into your stomach, it go into a digestive process and then go to your bloodstream. Normally get metabolized through the liver and then go to your body, go to your target place for it to have its effect. Now, obviously, if you think about that route, okay, then you're gonna talk about high dose because in your stomach you're gonna have some absorptions, and there's only only part of the drug can go to your bloodstream. That's called bioavailability. Now, once you overcome that, you're going to have to deal with the people have. So, for example, liver issues, kidney issues that affect secretion and also digestion. You also have to deal with like elderly people, young people. So what we called the special population. You also have to deal with the people probably with other drugs. If the people, it would be nice if people only take one drug for one disease. But the truth is, chances are you're going to have more than one disease. You have one drug and another drug. So there's a chance for so-called drug-drug interaction. So that's basically oral, locally given. Say, for example, KX826 give locally can overcome a lot of those difficulties. For example, we don't have to depend on the liver or kidney for secretion. At least for major part, we don't have to worry too much about drug-drug interaction because it's very, very limited if there's any penetrate to the system. So we can overcome those difficulties. Of course, not all the drug can become transdermal.
Yeah, I just add a little bit. Wilson has been given a really good, summary of the oral and the transdermal. I just want to add a little bit summary about that. Yes, so for example, the oral delivery, we have to really think about it because it's compared to the transdermal approaches, it's more easy to get absorbed in the bloodstream and also through the organs. So from the side effect perspective, of course, compared to the topical, you really give more or severe systemic effects, and which is beyond the target area. And also for the transdermal, topical drug, usually we need to consider the skin barrier and stability, and also, sometimes you have to consider of skin sensitivity. And so those are all in terms of the KX826 development process, we have to overcome all those considerations. So which means out of like tons of the compounds, we selected the KX826 and also GT229 are the best ones, and we have passed through all those tests during the development. I hope this information could help.
Yeah, thank you. That was a really good response. That's interesting. I guess when it comes to the discovery process, like, I don't know, like we hear today, like there's AI being used to sort of, find these various drug compounds. I'm kind of curious, what was the discovery process like with KX826? Whatever you can share about that. How was the drug discovered? Did you guys go through the literature first? Because there's other compounds that didn't necessarily make it to market, particularly RU5841 or PSK3841. If I recall correctly, that is a non-steroidal anti-androgen. I think you talked about this earlier, Wilson, but I guess more so specific to KX826, what's the story behind that? How did it come to an idea, and then how was it executed to actually go about, formulating the sort of compound?
Sure. Obviously, I was not an expert. I'm not an expert on this. However, and the development of this drug is way before AI. But the development of 826 is a structure-based computer assistant design. So essentially, we design a compound based on what we know. Look for a lot of literatures. We design about more than 300 molecules. One of the things we are leaning towards at the time was a concept called reverse prodrug. Of course, I think lots of audience probably understand what it is. Prodrug, the reverse prodrug, is a compound that is designed to be activated inside cells rather than in the cell as a whole. So that once, so it's referred to a molecule designed to initially be high permeable to a cell membrane. And once it's there, they can go into the cell and release active drug molecule. Now, one of the key things we try to avoid is systemic side effect. We also understand no matter how you design, there is a chance at the time the drug can go to the bloodstream, go to system. Systemic exposure will have some systemic exposure, and once we hope it was at the time is once the drug go to the liver, it can be metabolized to the less active AI inhibitor. That was a design concept at that time we hope to achieve.
Okay, yeah, thank you. Moving on to the next question. This more so involves the clinical trials, particularly KX826. So the first phase 3 clinical trial, if I recall correctly, I think that was December of 2022 the results came out, or December 2023. It's been a while. Maybe somebody in the comments can, remember that for me. But that clinical trial, if I remember correctly, was around 6 months long, observing KX826 at a 0.5% concentration, but it didn't yield statistically significant results against placebo when it came to hair growth. However, in the 1 year phase, I think it was phase 3B clinical trial, it did find statistically significant results. I guess what changed in this process, considering how it was the same concentration that was used in both of these studies? And what methods were adapted? Was there better patient compliance? I guess, what changed in that process?
Yeah, that's a great question. Unfortunately, we cannot mention this, explain this without talking about politics. So if you follow this drug on phase 3, I know lots of people have high hope on the phase 3 studies, and we are also very, very lean, have a good results. But if you look at the study, it's a 6-month study. It actually, from beginning to the end, took us 2 years. So, by the way, Kyle, the results came out exactly 1 year ago. So it's 2023. So
Okay,
it was
thank you.
2 years. It should be 6 months study. Normally, we're talking about from beginning to end 1 year, recruiting those kind of things. And I can tell you, AGA patient is not difficult to recruit at all. The reason for the 2 years is because, first of all, as you probably know, the COVID situation in China, that have a huge impact. We were forced to stop the study, pause the study, let alone to say other factors. I'm just going to list a few. First of all, compliance is out of the window. You really cannot check. We cannot even, for the people who need the drug, we cannot ship out because you just can't. Everybody is quarantined at home. But I guess once you are in a very stressful situation, worry about the food, and unfortunately, we think that the compliance or put those drugs on your hair is not a first priority. So itself is a very stressful situation. Stress can lead to some changes in your hair. Of course, COVID, stress, fever, disease itself can also affect. From our sponsor perspective, is that the compliance is really non-exist. So that's that. Now, what's the difference between studies? Obviously, if you look at time, so the second study is still ongoing. We haven't finished it. We will be finishing by the Q1 next year. Although that's 1 year study, you see that study starts at the end December 2023 is the time when China stopped the COVID policy, quarantine policy. So we actually, the most of the recruiting start after that. So I want to, of course, we pay attention to compliance. We have very good compliance readings. We follow up the patient. Those help. The product is the same. So I guess if you ask me, better compliance, better patient follow up. I guess overall situation. Unfortunately, the study results of a study like that in China is not only our company. The other company have the similar issues. If you have a very long study, unfortunately, that's what would happen.
Yeah, that actually makes a lot of sense, to be honest. Moving on to the next question. In the phase 1 study of KX826, it seems as if serum levels of KX826 were observed in the clinical trial participants at various doses.
Mhm.
There seems to be confusion from the public as to whether or not KX826 goes systemic, and to the degree in which it does go systemic, is it enough to cause side effects? What is the particular pharmacokinetics and pharmacodynamics of KX826, as well as its metabolite? If I think it's KX982.
A2.
And how long does it stay attached to the androgen receptor? That's another question people have. And if it isn't serum, if it does go in serum, do you know the approximate time it stays in serum? I think there were details about some of this in the phase 1 study. I vaguely remember seeing like a graph about the different concentrations and the peak time. But I guess some people, they don't look at all these things. There's a lot of studies. They don't look at all of them. So they want a somewhat of a clearer picture.
Yeah. Well, thank you for the opportunity. That's something when we do the Ask Me Anything, we also see the question because for the, we had to respond quickly. We really didn't have too much opportunities to explain this. So first of all, just let's just talk about this, how it can go. Okay. We would hope the ideal drug can only act locally, do not go to the bloodstream. There's no systemic exposure. That was the ideal situation. For a lot of those kind of local drug, that's the ideal situation. For our drug per se, here's what we observed. So for the single dose, use this single dose. And in all those different concentration, we didn't see anything in the bloodstream. What they call is below the limits of quantification, meaning that for the technology we have, for the way we measure it, we cannot see it in the bloodstream, the serum. Now, as when you go a little higher, in some dose, you can see something in the serum. So that's the fact. Now, the other thing is you need to determine how long the thing in the serum. So the concept here is half-life. So meaning how long it takes for the drug in the serum reduce its concentration to half of whatever initial concentration is. So, but here the thing, normally the standard is you need to have 3 samples. So for our drug at the therapeutic dose, 0.5, so what we have is about 30 or 40% in that. We cannot see anything, meaning below the limit of quantification. Of the rest, 30 about 60%, half of them, or a little bit less than half of them, we can only measure once. So we cannot measure second time, meaning at the second time point we measured, then it cannot see anything. So we cannot calculate the half-life. Now, then there's a 20-30%. We did see second times, we calculate based on that. Of course, we can, if you're talking about half-life, we can only say, those are measurable half-life. We can forget about the rest 70. For those about 20-30% of the patient who has a half-life is around 8 hours to 20 hours. Why such a big variety? The variation is because that's exactly what I just discussed. It's because of such a low number. It's it's variable. I hope it's clear.
Yeah, that's clear. That makes a lot of sense. In fact, I wanted to just add one thing too. I think also it's like patient habits are something to consider as well, because I know that people tend to microneedle with other products as well. So that's something to kind of keep in consideration. If anyone is out there, if you microneedle your skin, and I'm just talking to the audience, if you microneedle your skin, whatever you apply topically, you're probably going to get more systemic exposure too. So it's just, I've just seen this so many times in the community where somebody would say, "Oh, I got a side effect from this," but then it's like, well, maybe you did, but let's look at your habits, right? What else are you doing? Are you using things that increase, the absorption, right? Are you using a microneedle to, increase the absorption or the concentration absorption of a particular product that you're using? So maybe people have to be a bit, aware of their habits when it comes to, topical applications.
Great point.
Yeah, I think that's exactly what happened, Kyle. In following all the, we closely follow those, people's feedback, regardless what source it come from, Reddit or YouTube or, any sources. And we found, most of the cases, people not only stick on one, treatment or one product, they usually combine many products. Someone, use finasteride, even someone is working on the same pathways. For example, finasteride and the KX826 somehow is, they work on the same way. So in that case, like you said, they use different equipment like microneedles, micromassage. They combine everything. Sometimes it's really, difficult to say, what exactly and what kind of, affections with those combination applications. So that's exactly like what you said. And most of them, we don't have direct data to support, it's due to any products. It's just the real situations.
I think it would be good for people to report, if they do have side effects, but they should probably report what other drugs they're also using in combination with, the drug that they're complaining about. Just so, again, like, I think Wilson mentioned this earlier, but like when these things get to the market, there's usually some sort of surveillance that's still going on, right? So people obviously,
correct.
if you have any issue, you should kind of report it.
Yeah, we have a number for it in our website.
Yep, and I'll include that in the description, just in case if anyone's going to use KX826. But moving forward, though, sometimes when drugs bind to receptors, they could upregulate the number of receptors as the body attempts to, compensate for the lack of stimulation through that particular pathway. Has KX826 been observed to do this? And if so, does it risk limiting its efficacy over time? I know there were some people that were speculating online in the most recent 1-year phase 3 clinical trial of KX826 that there was some variation and decline in target area hair count, but although it should be stated that you have other factors like seasonal variation of hair growth, but I think just an important part of that is that the trial participants in the arm that was treated with KX826, they had a higher target area hair count than they did at the end of the study compared to the beginning. But just just speculating on this, is there a risk of upregulating the androgen receptor in the hair follicle by sort of antagonizing it?
So we are scientists. We can speculate. We also have some facts, and these are the questions. Very, very interesting. So let's look at the facts first. First of all, for us as a company, we do not have direct evidence. I asked before this discussion. I asked for our clinical group. They have no direct evidence. Whether this can happen? Well, first of all, if you look at our data, whether the 42 weeks is a little bit lower compared to 12 weeks. Yes, like 1 or 2 or 3 hairs less. But to explain that, there are other reasons. Most of them say can be caused by other reasons. But if you look at the Minnesota data, they peak at 16 weeks. We check all the similar. I can't say similar drug because I know there's only 2 approved. At least for both of. There's another drug is called CB0301 by Italian company. They are doing the study now. All 3 drugs, what I can say is they all have this peak. Whether it's 24 hours or 24 weeks or 16 weeks, it's around that neighborhood. It's all going down. So going down a little. So if you ask me as a scientist whether there's a possibility, I cannot tell you definitively yes or no. I can say these are the things that for the future research. So I'll leave it as that.
Yeah, I think that's a good point. I also noticed that with the CB0301, Breezula, I think that's the name for Cosmo Farm, but essentially it showed the same thing. There was a peak and then a decline. But I do think the difference here, I think their decline went well below baseline. But with KX826, it just stayed above baseline in your phase 3 1-year KX826 results. It did stay above baseline there. But there, again, I've looked at the literature too. I haven't seen any sort of evidence of androgen receptor upregulation, particularly in the hair follicle. Right? I haven't seen that. I've seen speculations of that occurring in like prostate cancer cells, but just understanding the pathology of prostate cancer, these are cells that want to feed off of androgens any opportunity they can get. So I guess the evolutionary mechanism of a prostate cancer cell, because of that, the need to develop from androgens, any kind of androgens, it makes sense that in its sort of DNA, it would kind of develop the mechanism to upregulate androgen receptors. So when I hear people online, arguing, oh, these inhibitors or whatever, they these antagonists or whatever, they're going to cause upregulation, and then they point to like a prostate cancer cell. That doesn't really make sense because these are two different cell types, the hair follicle cells and the prostate cancer cells. So that's just one thing I wanted to kind of point in there to the audience. But moving on to the next question. Much of the press from Kintor highlights KX826 as an effective alternative to finasteride and especially dutasteride. However, these drugs have been on the market for years with proven efficacy, especially, in the long term. So why would somebody pick KX826 over these drugs? What are some cases where somebody would say, oh, I'd rather go with KX826 rather than finasteride or dutasteride?
So that's a great question. So I think we're talking about marketing position. I think in the end, KX826 is another drug on the market. It has different MOA compared to other drugs, as you mentioned. So consider finasteride. We are locally applied, and we can avoid a lot of side effects. I know we're going to talk about a little later about side effects. Compared to minoxidil. Minoxidil is a very effective drug. Of course, it has been in the market for a long time. It was developed in the 1970s, I guess, and been in the market for a long, long time. It has its own characteristic. It has their own safety issues or sheddings. People shed. There are different philosophy, different theory about the shedding. Lots of times they can shed. The hair can go back. Lots of times they may not. Lots of times people hate the experience of shedding. It is an effective drug, but not all the people can benefit from it for whatever reasons. Now, for this is one additional drug on the market for people to choose. It has its own advantage. We saw some shedding, but it's not as big as a minoxidil, mainly from the post-marketing. We have a pretty good safety profile, and those are our advantage. One case I want to make is that if you look at the MOA, we're totally different. So we are actually explore for some small studies, try to see if you combine these two drugs can have a synergistic effect or at least additive effect. Understand there's of course a limitation on that is that both drugs are used twice a day. So that's a hurdle we are sort of try to overcome, and I hope I have an opportunity later on to explain why this 20029 come from and how that hopefully can solve this issue.
Yeah, that's a really good point. Really, really good point you make there. The next question would be, and this is in regards to side effects, KX826, as phase 1 has shown us, I guess does indeed go systemic to some extent, depending on the concentration. But with this sort of systemic availability, in some people, side effects, who knows if it does occur. Some people have nocebo effect where as soon as you tell them side effects are possible, instantly, they'll think the worst thing is going to happen to them. But considering how this drug is designed to block the androgen receptor, if there is side effects, would these side effects be similar to those seen with finasteride and or dutasteride? Because with finasteride and dutasteride, you're just blocking, the 5-alpha reductase, but with a sort of, antagonist to the androgen receptor, you would be blocking the action of androgens theoretically. So would there be a similar side effect if it is to happen?
Okay, that's a great question. So I can share with you some of the data of the side effects that people are paying a lot of attention to. I will tell you what is firm, what is not firm. When I say firm, means that the study is done. So we can say that, well, we have this and we have that. And not firm is that I'm going to cite some long-term safety study. As I told you, long-term safety study hasn't started, just hasn't ended yet. So things can change. So basically, And then we can speculate what can happen. So on theory, that can happen. Of course, if you expose the body to enough of those AR blocker inhibitor, things can happen. And then if you think about it, adverse event, a lot of times, as you just mentioned, especially in post-marketing section, so people, it happened. For example, I know people are very focused on the sexual male dysfunction, whatever sexual related malfunction. That things happened in real life as people, men or women getting old. These things happen. There's a lot of literature. I'm sure you know that cited the percentage, like situational, like 40%. It could be higher, could be lower. That's part of life. Now, what can we do is that we, in a controlled study, we have a comparison. We compare with the placebo to identify a potential adverse event. But the problem with that is that we're talking about limited number of patients. Think about it. Well, in a clinical study, we have like 400. We think that's a lot. Well, in the real life, we're talking about 400 million people may use this. So whatever you count, that may or may not work. As most importantly, some of the adverse event is not going to happen in a small number of patients. So that's the reason. If you think something happened, give us a call. We have a number that we'll try to get those information. That's our obligation. Okay. So having said that, let's go through the 1003 study. So 1003 study was complete. So we're talking about 740 patients. Half of them on placebo, half of them was on 0.5% KX826. There is only one sexual related dysfunction side effect was reported. That was ED. Fortunately for us, that patient was on placebo. So that's the only one we find. That's the data. Whether that can happen more, well, we can speculate. In 1005, so 1005, we have about 200 patients. That's 1 year study. We do have one self-report sexual dysfunction. The intensity is mild. That patient did not seek medical treatment. With the same note, the patient also report that patient has shingles at the same time. So that patient sort of experienced painful rush in the body. At the same time, he felt that the adverse event report as a sexual malfunction. So that's the only two things happened. Combine those two studies that's the only thing happened. Reported side effect AE in our more than 1,000 patients treated with 826. Of course, so as I said earlier, if we go to the market side, there things can happen. We're looking forward to getting your report if there's something bothers you, and do analysis and try to see if that indeed can happen. Though we do not have too much reason to believe that will happen, because for MOA, from the limited exposure, but things can happen.
Yeah, thank you. And again, that sort of details, if you guys do get KX826, will be in the description of this video and also the comment section. So moving on to the next question, are there any interactions that you guys have seen between topical minoxidil and KX826?
You mean the interaction between minoxidil and the KX826?
Yeah, are there like any negative interactions between the two chemicals?
Well, if you think about it, both of if you think about locally, at the moment, I cannot tell you for sure, because as you know, we're doing a study including 120 patients. That combination is a compare with minoxidil plus KX826 0.5% versus minoxidil. So that's the study we'll conduct. It's 120 patients. Data will be available sometimes in late Q1, early Q2 next year. So far, we did not see any adverse event report that make us think there is a possible side effect if you combine those together. But of note, there are a few things that if people want to do that, people need to understand a few things. First of all, if you think about systemic exposure, both drugs are pretty low. The chances are you're going to have systemic exposure and then have drug-drug interaction as a normal sense is extremely low. Locally, if you use those two drugs, if you just follow the direction, you're talking about, 4 times application. both of them is like 2 times a day. So if you follow the instruction, it's 4 times a day. Whether there is a local reaction or allergic effect, it's possible. What I can share with you is in our study, conduct about those 120 patients, we instruct the patient to use, for example, minoxidil and rub it for 5 minutes, at least 5 minutes, before you use KX826. There's no, we don't think the sequence has any effect. Another way to say it, people could use KX826 and then minoxidil, but we ask that patient use it, rub it, then wait for at least 5 minutes before you use the other one for precautionary. But so far in clinical study, we didn't see anything in terms of side effect.
Thank you for the clarification. That was something that some people were, I guess, speculating on online. So it's good to kind of get some more information on that. The next question is...
Yeah, we'll provide more information when we have the data so that you can see the data. But as I said, the study is ongoing. It's very hard and everything has to be verified.
Yeah, I agree. The next question would be, is it a practice in Kintor's clinical trials to perform biopsies to confirm androgen receptor density throughout different regions of the scalp? And are biopsies used to confirm if KX826 is indeed acting on the hair follicles or if it's saturated in, specific areas? That's
a great question. That's something when we talk to the regulatory agency, and a lot of people suggest we do that. We were thinking about doing that in our early studies, but let me say when we try to do that in biopsy in this, we got a lot of protocol rejections from investigator sites and also patient. They just don't think AGA is worth of doing this biopsy. So the answer to that, at the moment that we're trying, we haven't got the data. However, in the study that we plan to conduct in the future, especially 20029 in America, in other places, we'll definitely do that.
Yeah, that's a good point that you bring up. How, the investigator sites still, a lot of people want to have biopsies done. Even here, we don't have enough biopsy data for conditions in general when it comes to other like hair loss disorders, because biopsies just aren't frequently done. And in the other pharmaceutical companies that I've, kind of observed, they don't seem to be doing biopsies. I don't even think it's like a standard practice to do biopsies in these studies. So it's, it's good that you guys are, kind of looking for it. Yeah, it's very, very hard.
KOLs, the question we ask is whether it can be done. The answer is yes. Whether it should be done. Well, the answer from some KOL, American KOLs, is it depends. First of all, what you try to say? Try to say that the binding, whether the binding correlate to efficacy. Well, it's hard to say. What time point you're gonna get? So if you give the drug, you have to choose a time point to get the biopsy. Then ideally, you want to see at the time point, I got a biopsy, I see the binding, I see the interaction, then we have a result. So it's very hard to determine these parameters. It's just, close to impossible. But if you want to just see the in vivo data, in vitro data, that's probably okay. But that's the reason even the KOL has different opinions sometimes.
Yeah, I completely agree. It's, you do have to set up those endpoints, and for some things that are very minute like this, like if you set up an endpoint for like 2 months, I think I saw this with dutasteride by the other 5-ARI drug, where in some studies they only check scalp DHT at specific endpoints, right? But in one study concerning, I think it was concerning people that had either benign prostatic hyperplasia or prostate cancer, they chose to have the endpoints every 2 weeks, specifically testing tissue DHT concentration in the prostate. But, who's to say with the study that only checks scalp DHT concentrations, what if they checked it every 2 weeks? That would be, a lot of biopsies to do on every subject, but you would certainly probably get different results. Maybe it would tell you that the serum concentration DHT levels don't take necessarily a month for them to reduce, but rather it's just the endpoint was, you do have to have an endpoint. It's kind of hard to determine what that endpoint would be.
Yeah, that's great comment. So that actually for the audience, I think that that's something about the pharmaceutical industry and also basic science. In basic science perspective, you want to know everything. In real life, well, there's other thing to consider, ethical consideration, this consideration, and whether the patient is willing to do this, and also what are the results that can be helpful. It's certainly not ethical to just say, hey, this is something in research question I want to know, but without well planning. And also you have to think about, well, what if the data come out this way, how to explain this? So those are the things we consider in the human clinical research. It's not that something that we want to do, we can. It's just there's a lot of limitations.
Yeah, I don't think people really recognize that, because they ask questions like, okay, why isn't this being done? Like the biopsy thing that I, brought up. Although it would be really nice to get that sort of data. If you don't have a significant amount of patients that are willing to, have a part of their scalp cut out, then if you don't have a significant amount of patients to do that, then you just can't, do it or get that much ample amount of data for that to be the case. So that's a really, important part that you bring up. Moving on to the next question. This is something that everyone has been very interested in, GT229. This is a lot of people want to know more about this, as much as you can share. Can you explain, just to the audience, to the uninformed, how does GT229 work? Like, what is this?
Yeah, I can tell about this, just from the mechanism perspective to help the audience to understand. So the technical use for GT2029, which is called PROTAC, is PROTAC. So in general, it's referred to a protein lysis targeting mechanism. So instead of directly binding like KX826 or the other, topical like DHT blockers, GT2029 is using technology related to the protein degrading. So instead of directly binding, it's actually help or navigate the protein to degrade. So of course here the target protein is androgen receptors, which is root cause and the main theory of the issue of the AJA. So with the specific design, GT229 can capture the androgen receptor and recruit it's called E3 ubiquitin ligase, and it can active a protein system like a ubiquitin proteasome system, and which is basically like a cell's garbage disposal. By recognizing the androgen receptor protein and can completely break down through this system. And for GT229, it's designed for topical use and it can selectively degrading functions on androgen receptor. And in that point, can reduce the androgen receptor within the follicle and, of course, limit the cellular functions. So in that way, of course, reduce the DHT impact. And since it's topical use, so it has very limited, actually alerting the other systemic hormone levels. So compared to the directly binding, which is temporary and reversible, and that's why it's need frequent use. Basically, from current real world situation, all the other drugs you have to use daily, once or twice a day. And so this, by using this protein degrading mechanism, can achieve long-term reduction in protein levels and activity. That's exactly what we have seen in the clinical trial settings as well. Later on, I think we have opportunity to just give a little bit data about the GT229 clinical trial results. So I just want to mention, so PROTECT technology has been gained a lot of attention in recent drug development, especially in cancer research. However, GT229 is the first topical AR PROTECT compound and entering late stage in the clinical globally trial settings. Yeah, that's the background mechanism to understand 2029, and this is a little bit comparison with the other with the other DHT blockers topically used.
Thank you so much, Grace. Moving on to the next question. How often is GT229 theorized to be used? So currently, what is the current frequency of use in clinical trials?
So, as Grace just said, there's totally different MOA. The other talk about competitive inhibition. We're talking about twice a day, and the limits of that is that, first of all, who can honestly, I would challenge everybody, said that over the past 6 months you use it twice a day, every day. Even if your best intention, there is a chance you'll miss. Maybe you just go to a business trip, maybe you just have a Sunday night, Saturday night party, you forgot. And the key of the GT20029 is based on animal studies. This degradation happened and maintained for 7 to 9 days in animal model. Obviously, we need to verify that in clinical study. In our clinical studies, we use 2 dosage. One is once a day. One is twice a week on the first and the fourth day. So we find those are similar efficacy. So in the future, we're going to use in our proposed phase 2 or 3 studies with our government, we're going to use twice a week dosage on the first and the fourth day. So that's that for now. Hopefully, once we get this done, we can explore even longer or different variety of those frequency of use.
Yeah, thank you so much. That a lot of people, I guess when it comes to compliancy, it would be nice to have something that you don't have to use every single day. And I think a technology like this kind of hits the sweet spot where it's not something that you have to maybe, I don't know how it's going to turn out, but it's not something that it seems you have to apply every day, like a topical minoxidil, So it's definitely pretty interesting tech. Moving on to the next question. What was the reason for using, and maybe this is just my understanding, but what's the reason for using a gel formulation of GT229 instead of it being distilled in a solution like KX826 is?
We have a gel and a solution for both 2029 and the KX826. It's just different the indication. Gel is for acne, and the solution is for AGA.
Oh, okay, I understand. So, okay, so there's a gel KX826 or gel GT229. It's just that's for the acne sort of, I guess, trials, and then there's the solution definitely for the. Okay, cool. Yeah, we rarely hear much about the acne trials, though. I mean, maybe it's just due to bias because everyone's mostly concerned about AGA. Just real quick, what's going on with the acne trials for either GT229 and KX826?
The KX826, we had a trial about a while ago, and while it's efficacious, we believe that we can use even higher doses. At the moment, we are thinking about conduct high dose of 286 for the acne. For GT2029, we'll conduct 165 patient study as we speak. It's a double-blind study. It's double-blind studies. We have two doses for the GT2029. Try to first study of proof of concept study is ongoing. We have about 65 patients enrolled. A little slower than we anticipated because we need the PK data. Hopefully nothing there. So it will be finished by the end of next year. That's a 3-month study, typical for acne.
Okay, yeah, thank you so much. So now it's going to be the sort of kind of a mirror of the KX826 questions that we had before regarding the pharmacokinetics and pharmacodynamics of GT229. Maybe you just touched on this a little bit, but I'm going to ask the question. As it currently is understood, what are the pharmacokinetics and pharmacodynamics of GT229? And has it been detected in serum? And if so, if you guys even have the data yet, what is the sort of PROTAC's half-life? Because this is very new technology. People don't really know what a PROTAC is. Grace, eloquently explained it. But I think people would like to know, like, what is a half-life even regarding to a PROTAC tech?
So Grace can add this. I can tell you the half-life cannot be calculated because most of the sample we got is below the limit of quantification. So it's to a point, it's certainly more than 70-80%. So the half-life cannot be calculated. Grace, you want to add anything?
Yeah, that's exactly the situation. We have been, communicate with our research and development department, and so it's most of the samples, since it's local use, so most of samples is undetectable, and which is also means less like 10 picograms per milliliter, and as too many samples are undetectable, it's not able to accurately calculate the half-life, and we don't want to give this number, if we don't have the exact numbers to be misleading on the audience. But that's something we are keep pursuing and trying to understand better. Yeah.
Okay, yeah. Thank you for that answer. The next question, for GT229, which targets the androgen receptor by way of the PROTAC technology, how does its degradation mechanism differ from other androgen receptor inhibitors in terms of safety, efficacy, and potential for resistance?
Yeah. So as we talked, at the beginning, the topic of the GT229 mechanism. So 229 actually recruits E3 ubiquitin ligands to the androgen receptor protein for ubiquitination and degradation. And the degradation affects what we have observed in 7 to 10 successive days, still in the in vitro studies, which indicates that GT229 could be applied in low dosing frequency. And this topical formulation really is limited to systemic exposure, reducing the risk of the side effect, and such as hormone imbalance or the other issues commonly seen with the systemic AR inhibitors. Yeah.
Thank you for the answer. Now, this kind of goes into your pipeline at Kintor, right? So what would be the purpose of developing both GT229 and KX826 if they both have the chance of being effective? Is there a plan to use these drugs together?
If you look at the MOA, you can use the one is the AR protein receptor degradation, the other is inhibition. Normally, one is hopefully can be, at least for AR 2029, we can be degraded. So I don't think it's probably a good idea to use it in conjunction, especially both of them are topically used. Why we develop two? Well, I think the 2029 is with the compliance, of course, and convenience for the society in mind. As I said, we did a lot of research, and also people, even in my company, are tested themselves, myself included, whether it's feasible to use a drug twice a day for a long duration. It's very, very difficult if possible. So with this GT2029, it will make use drug more convenient, and convenient, and in that regard, the compliance will be high. As technically speaking, if you use 2 days a week, sort of there's something in your back of your mind, and when you think about it, you should be able to met 2 weeks a week, 2 times a week easily. Obviously, very lower, or if any, I can't say if any, very low systemic exposure is a plus. In our study, although we just had a very first phase 2 study, as you can imagine, there's really nothing there. That's just verified this low systemic exposure. So that is our exposure. Certainly, 20029 is certainly is our focus in the after 829. We are at the moment working with the regulatory agency to design a phase 2 and 3 studies. Hopefully, we can start sometime in the mid or late next year to get things ongoing.
Yeah, thank you for the answer. I guess the next question, just taking a bit of a step away from GT229 and KX826, because these, drugs that you guys are developing seem to be very applicable to just androgenetic alopecia. Is there any other interest for Kintor in looking at other forms of hair loss, such as alopecia areata, or even scarring alopecia conditions like lichen planopilaris? There doesn't seem to be much on the market regarding that particular condition.
Yeah, because when we talk about AGA, androgenic alopecia, so there is certain reason on that, and why GT229 and KX826 work on those. It's basically AGA. Beside the genetic reason, there's one main reason is about the DHT, malfunction. So, but for the others, such as you mentioned in alopecia areata, that's different mechanism, that's different reason. Mainly it's because autoimmune system, the abnormal. So that's why we do not, and from the mechanism perspective, so for that alopecia areata, we do not think those two compounds would work on that. And in terms of the other, indications such as, the oil control, that's the main things we have been observing from the any subjects who are using our products, especially for KX826, we have more data, accumulated. So we have observed, very strong oil control functions, and that's also, from deeply understanding DHT, mechanism with the DHT, the function, abnormal function. It can also, work in the follicle and stimulate the oil, secretions. So from that perspective, KX826 has shown very excellent, function on that. And from the other perspective, associated with alopecia, sometimes we will also observe some people have like inflammation conditions. That's what we are starting to, accept, is starting to receive the feedback from the subjects who are using the Cochin. They found they are scarring or, very persistent of inflammation in the scalp has been slowly to get improved, and that's what we believe, KX26 from the mechanism and from the performance so far in the market, not only be able to to help or slow the hair loss, but in the meantime can have very good oil control and somehow can do the inflammation control as well. That's from the data and observation so far.
Yeah, thank you. That makes sense. From what I know from like other, let's say, scarring alopecia conditions, it seems to involve a defect of the PPAR-GAMMA receptor. such that, when this receptor isn't working properly, and maybe the same with the retinoid receptor, it leads to a buildup of, lipids in the tissue. And ultimately, the immune system is triggered, the lymphocytes are released, and they attack the sebaceous glands and they destroy them. But another interesting aspect that you brought up, Grace, and this has also been noted in other pharmaceutical companies. I think Wilson also brought up, Cosmo Pharmacy. So far, they only have, I think it's called Winlivy, that's on the market. But we know this from the literature. DHT, as you guys stated, stimulates the sebaceous glands to produce, more sebum, right? But I sort of like see this, this intersection between as an individual that might have some sort of polymorphism or defect to their PPAR-GAMMA receptor, such that when DHT overstimulates the sebaceous gland, it produces excessive amounts of lipids. And because PPAR-GAMMA is involved with, breaking down these lipids and metabolizing them, if that receptor is defective in that case, you by way of, DHT stimulation or 5-alpha reductase activity in the sebaceous glands or their sebocytes, you end up having an abundance of, sebum, of these lipids, sebum high in cholesterol and triglycerides that ultimately can, cause for various conditions that are very inflammatory. So yeah, I do think, like, just my personal opinion, with KX826 and, its efficacy in oil control, right, limiting that excessive amount of sebum, it possibly could have some sort of indirect, action on, other forms of scarring alopecia, right? And I've seen this in other studies where, they would give dutasteride or finasteride to somebody that has frontal fibrosing alopecia, and then they see an indirect, observation of where, okay, there's not as much oil anymore, and because of that, there seems to be an improvement in hair counts in specific areas. It's not going to be the best, but just, my opinion and how I've seen the literature. I think there's maybe an indirect kind of benefit to these sort of products.
Yeah, that's exactly. To have the hair loss or androgenic alopecia, that's a combination of a lot. It's a complexity, reasons working together, including the excessive oil secretion or, the inflammation. So by improve any aspect of these conditions, it can, just rebalance the health, hair, growth environment. So, and eventually we hope it can, work on the androgenic alopecia. Yeah.
Yeah, this is the great point. I think that something you mentioned, we already bring back to our basic science team to look into it. Just for this oil control, I think that I can tell you that there is talk in the market, because these are like we know on concept. Those are possible and feasible. However, in clinical studies, that is very difficult to measure. But the thing is, and we also have a set of inclusion and exclusion criteria, as I said. So as I said earlier, there are utilities for clinical studies, preclinical study, clinical study, and post-marketing. In the post-marketing, as you know, we're selling this compound globally. In China in particular, we got a lot of feedbacks that if you have oily head, this drug works perfectly. So much so that people. So it's can we just specifically give it to those people? We have strong evidence. However, that's remain something to be seen. How the patient can feedback, patient's feedback.
Definitely, yeah. Very interesting topic. Just this alone is very, very interesting. But in regards to the patient feedback, how does Kintor handle feedback from patients in clinical trials, especially regarding side effects, ease of use, or general satisfaction with the treatments, right? Have patients' reported outcomes influenced by the development team? Like, maybe let's say a patient gets, a side effect, or they complain about the let's say propylene glycol, right? In one phase of the study. And then do you guys take that into consideration for the next phase of the study? Maybe instead of using propylene glycol, you might use glycerin or some other, humectant.
To answer that, yes. the formulation has been changed throughout the drug development process. As a matter of fact, we're looking for the third change. In the beginning, in the phase 1 study, as you expected, there's a lot of side effects related to that. So we hear when they respond, we see the adverse event, we see the feedback from the investigator. So that's something we're improving. To get back to your point, I think that in let me just say this: in clinical studies and in post-marketing, in clinical study, we as a sponsor do not interact with the patient. It is our investigator interact with the patient. That's part of the GCP. So basically, they report whatever the adverse event or feeling, whatever, to investigator, which then give to us. Patient will be evaluated for the patient report outcome. One thing that people have to understand, there's a difference about the clinical studies and patient report outcome. In the clinical studies, we have the sort of target area, so we measure how many hairs increase. But the patient report outcome is different because as you can probably see, that if you talk about your own hair, right, it's very, very difficult for you to have an accurate assessment because hair loss, AGA, is in part of the head that you cannot easily see. Even if you have 2 mirrors, it's very, very difficult to see or carefully measure. But patient self-awareness or self-confidence or what they perceive is very important. In our long-term study, we have a 60% patient report satisfactory or very satisfactory. So in our long-term safety study, so that sort of certainly give us a lot of confidence and hope that we can do something to help those patients. But on segues to the people that listen to this, I think that AGA, unfortunately, is I won't say the disease, but at least it's a chronic condition. It is a condition a lot of people associate with aging or whatever. And when it's very, very difficult. It's very, very difficult to use something. We're aware that's the reason we developed 20029. It's very, very difficult to use something twice a day. However, the appearance is very important for all of us. And then something you're gonna have to try to stick, and it's a lot of time. It takes time for the drug to work, especially for the competitive inhibitor. You're going to constantly use it before you can show some efficacy signal. So just be patient and use it consistently.
Thank you. That was a very, great educational point you just gave the audience, Wilson. Because people don't seem to know, first off, what an investigator is. Sometimes they just think it's all the details just go right directly, like it's somebody directly from Kintor that comes in and does all the counting. But I think, people just don't seem to know what an investigator is, right? So that was a really good point that you kind of clarified for the audience. I've been trying to do that many times, but for whatever reason, some people they don't seem to like take that into account, right? Like these studies that pharmaceutical companies do, the multi-center, double-blind, placebo-controlled, randomized studies, it's very robust and it gives you some really good data. And the part that you bring up where you have the investigator assessments and the patient assessments, that's also another factor, right? It's hard for a patient to know, what's going on the top of their scalp. So a great point you gave to the audience, what an investigator is. That's a really great point.
Yeah, a lot of times the patient doesn't agree because, you have a device on top of your head, they're magnified 20 times, they see those things. And also, it's the investigator and study coordinator over there that's neutral. But you can imagine, if you have the things, have the sponsor, like us, Connor, if we know the results, we obviously have a bias. No matter how you pretend to be objective, they are also human. We are all human. We have bias. That's the reason per GCP regulation, is the investigator who's doing the counting. Investigator has to be trained and certified by government. So as you mentioned, it's very robust. So that's the reason people pay so much attention and weight on the clinical study results versus post-marketing. In the post-marketing, it's an in-control situation. I will argue lots of people think you have AGA, may or may not have AGA. You may have something else going on. And even if you have AGA, there are other factors can influence your hair. For example, stress, for example, have a fever, or have a COVID, have other conditions. Nobody knows. So then you use this drug, it may or may not have the efficacy you are hoping for. That doesn't mean the drug is not working. It just means there are other factors. In clinical study, it's more objective. And if you, especially talking about long-term safety study, when we sort of follow the patient every month for 12 months at least, to look at every angle, to do all the testing, draw the blood, do observation, talk, and have a lot of self-report results and compare with the baseline. Those are where the results come from.
Yeah, and I just want to say another good point that you brought up was the fact that, yeah, if you don't especially with hair loss, because sometimes you can have hair loss that, looks like another form of hair loss, right? If you have AGA, you could have another condition that's mimicking the look of AGA, right? So it's like people have to understand, these studies, at least in regards to the kind of hair loss we're talking about, it's androgenetic alopecia, AGA. For those of you who don't know what I'm saying when I'm saying AGA, that's what it is. So that's a good point. People have to be mindful of that. If you have some other condition like pseudopalliative Bruck, or let's say you have a hair loss due to a traction alopecia, you can't expect a drug that works on treating AGA to completely work on maybe something that doesn't even have the target mechanism of action for that particular condition. So it's one point that people have to get keep in mind. But yeah, I guess what other question do I have here? Yeah, what's the delivery of GT229? Is it in some sort of nanoparticle, or what's going on with that?
It's a solution. It's the same thing, same as a 826. We're exploring other ways in conjunction with the others, but at the moment it's a solution.
Okay, yeah, thank you. Now we're going to move on to, I guess, when it comes to the accessibility and the pricing of these sort of products or these drugs. Regarding sustainability and cost, how does Kintor plan to price? I mean, you already have KX826 out as a cosmetic, being the Koshine 826, if I recall correctly. How do you guys plan to price maybe at higher concentrations KX826 and perhaps GT229 to kind of get the the happy medium of the affordability for the patients worldwide, while also maintaining your profitability at the same time to further innovation and other research?
So I think that's a great question. Unfortunately, something I cannot answer. It's because I'm a science-based, and I think we have the sales department to do those calculations. But it suffice to say, from my perspective, there are a few things to consider. One is the easy-to-use accessibility for the general audience, of course. Second, we also have to take into consideration the developmental cost. As you probably see, doing a research and do a lot of study, use thousands of patients, and also a group of, we have like 200 people work on this, isn't easy. So these are something we definitely have to take into consideration. I would think that we will certainly work with a different group of people and understand different region and keep the accessibility for each individual in mind. I know these are popular drug. If people like it, they can use it for a long time. Sign us, if you're really interested in this, I can talk to our department, pricing and also sales department to get some more clarification. At the moment, we only have a few products. By the end of this year or early next year, we have a few, including acne and formulation, and also have other things. At that time, we'll probably have a much better grasp in terms of the pricing. But the other thing is, as you probably know, we are really not a big pharma company in China, so we are still learning the pros and cons of each country. Try to understand more. I have some background with the United States, but we also operate in other market. So any feedback, any suggestions, anything are all welcome.
Yeah, definitely. And thank you guys for being like so responsive and like collaborative in this. Like, it's very rare that pharmaceutical companies and or biotech companies are trying to make an effort to like reach out to people. So and I've seen Kintor is like one of the very few companies in this space that actually makes the effort to do that. So I think the audience appreciates it. I certainly do appreciate both of your time. I understand for you, Wilson, it's very like early in the morning where you are. So
4:30.
4:30.
That's okay. I like the discussion. I want to ask you. Well, you just cut it off when you do that. What is your background? I'm very interested in learning your background. The reason I'm asking the question you ask is really insightful and very good depth. And then, a lot of questions are something we are struggling and thinking about it almost every day. And I assume you are not in the pharmaceutical industry. Am I correct?
Yeah. Well, so I'm a software engineer. Particularly, I do DevOps. So I know, yeah. I'm I'm not even in the same field. But in my undergrad, I was trying to pursue bioinformatics, but then that was at least a lot for me at the time. So I decided to just go the whole IT, software engineer route. But it was when I started dealing with the condition, androgenetic alopecia myself, I got back into like looking at clinical trials and looking at the literature, and I kind of became obsessed over it after a while. And I tried to apply like specific troubleshooting frameworks that I would gather from my job and just from, when I go about developing software. And I applied that, and I thought of the human body as this like complex, machine of some sort, and how I would be able to troubleshoot it. So, when I close my eyes and I imagine a receptor and what it does, particularly what I've been obsessed about recently being the PPAR-GAMMA receptor and lipid metabolization, and what that implies when DHT interacts with the sebaceous gland, all of that, how that sort of connects to each other. It's in my mind, I'm kind of like trying to troubleshoot and understand at a higher level and a low level of what's actually going on. So essentially I just read a lot. I'm not necessarily a scientist like you are, but I just read a lot. Yeah.
That makes us will work even harder. I think that some of the questions normally when the Grace told me there are some questions that I was like, say, okay, well, I let people how much they can ask, and I look at this question. Wow, I share some of the questions with our preclinical folks, with other folks. They were, wow, this is really a scientist ask a very tough question. So great. I mean, I think I like those stimulation. I also like the fact that you can bring some feedback from the market, from other people you hear, and give us a lot of like some of the questions they might be needing and how you can you use together. These are something that we sort of take your suggestions, and we'll try to work with the team to think about in that. That's great. And the personalized medicine, as you said, the question 32, that's exactly something we're trying to do. We're doing actually. So we're trying to sort of see what patient. It's not helpful it's probably a good idea to identify AGA patients a lot. It's a very, very tremendous big market. If we identify a subgroup of people that can use very efficacious, you'll actually do a lot of good things. I mean, make patient happy, make people happy. Because of that, such a big market and so many CNC influences, it also means that there were people not happy with the drug for a lot of reasons other than the drug. So that's something, personalized medicine is something really, really good, and it's something that we're going to try to see whether there's some aspect we can do to sort of narrow down or narrow the patient population so that we can achieve better results. Go on. Sorry, I'm just carry over. I think that you're great.
Oh, thank you. Yeah, I mean, I think that's it's a really good thing you guys are doing there at Kintor, to be honest, where, like I certainly see the effort and I see the work that you guys are doing. It's fantastic. It's actually, and honestly, I think Kintor doesn't get that much credit as it is, at least when it comes to like Western markets, because there aren't that many companies that are trying to innovate in this space. Like when it comes to a PROTAC degrader of the androgen receptor, to me, that sort of mechanism of action holds a lot of promise because, well, if there's no androgen receptor for the DHT or other androgens to, bind to in the hair follicle, then presumably, as we know from the, the sort of, the pathology regarding AGA, the hair should be fine. It should be fine. It shouldn't get any sort of negative effects from DHT at all. So, yeah, I certainly see Kintor as a leader in this space, and yeah, you guys are doing great, doing some really important work, and I think the audience and especially myself certainly appreciates that a lot.
Absolutely. Thank you so much. Yeah, thank you, Kyle, to provide this unique opportunity for us to be able to has the window or channel to communicate directly to help people understand, from the disease, from the condition itself, age itself, mechanism of the products, and also to understand, any products. For the position of any product, we do not, want to replace any, therapy or, current available product. We want to provide it an alternative, choice for people who is struggling in this condition, and so respect the product, the drug development, pathway, and also respect the hair growth itself is how have the cycles of the hair growth. And so that means, when you using or apply, especially for any new products, you probably need to give a little bit long time, to really to understand, hair is not, it's not like something you lay on the bed and you're gonna have the full head of the hair the next day. The hair do have its cycles to growth back. And for any new products or new mechanism, just give a little bit patience for trying and just be expecting the results, and compare with the other choices and to choose the best one for you. For example, for minoxidil, I think the number is only 10% of the people who suffering or the AGA can be stick on minoxidil, even it's minoxidil is one of the two approved from the FDA perspective. But for with the many products available in the market, our product is trying to just provide an alternative choice for those people. Since the AGA people population is really big, we just want to to do something and help the people who is eager to have different choice and different try. Yeah,
most definitely. And before we end this, I always like try to end it on something. You guys are well established in your careers. Could you give people advice, maybe somebody who, wants to get into, research or whatever? Could you give them advice about your space and, just like what you guys went through, how you came to your careers, and just general career advice? Because, sometimes I get comments, someone asks me, hey, so, how do I get into the pharmaceutical industry? And I'm like, well, I'm not in it, but I'm talking to two people who happen to be in it. So I just want to hear your response to that.
So I can go first. I think it is hard. It is not for a fake hard. And I think it's, I've been doing this for like 20, 30 years. I was lucky enough to be part of some very successful submissions. But on the flip side, I involved lots of failures, like 20, 30 of them. It's difficult because in the end, human body is, as you just mentioned, is very, very complex. And you think about you know something, but the truth is you don't. So I think, but it is a job that is very, very important as mankind. Think about 1940s something at that time. When we had the war, if you have infections, you're almost, going to die. But with the drug development, right, there are nowadays, if you have infections, you barely just need to do it. Just get some antibiotics. Most recent example, cancer. You probably know about 10, 15 years ago, if you have a lung cancer, you say, hey, maybe you have a year or two, maybe that's it. That's very personal to me. Well, my family, my dad, because of the lung cancer, passed away. But nowadays, with the new development of a lot of drugs, as you probably heard, Merck and BMS as this PD-1, the immune inhibitor, that make a lot of disease seem to be uncontrollable disease manageable. And sometimes you can have extend a life for like a few years, 5 years. Nowadays, you'll have lung cancer 5 years, almost certain. So those are because of a lot of us work on different aspects for this. So I'll say it is hard. It's a great work. But how to get in there? Well, there's a different path to getting to pharmaceutical industry. There are, first of all, preclinical side. Preclinical is something that you do a test. Once you have the design or compound or of the drug, you do the test in the animal. You do first of all, you do in the lab. Try to see whether this thing under some condition can work, right? If you have a drug that can lower cholesterol, you do the animal study, like for KX826, for example. We have lab scientists do these things on animal models that we get rid of animals' hair, then we use this drug and this can be a regrow, that per se. That's a lot of work. Those are the people that a lot of people will have masters and PhD. Those people deal with what they call bench scientists. They're working alongside the bench and they're working on different aspects on this. Then we have clinical person. Of course, when we do the submission to the FDA, we have a regulatory person. Those people normally have master or PhD, have some experience and deal with regulatory. They have to understand different laws. A drug can be approved in the United States. There's an ample example of drug approved in the United States cannot be approved in Europe, vice versa. Then we talk about clinical. Clinical is very complicated. If you think about animal study, one aspect of animal is under your control. You give animal food at 5 o'clock. That's 5 o'clock, right? But in the human clinical study, there are different, very, very difficult to control. Now, there's other people can get into it. The key players are like project manager, and as Carl said, there's a clinical pharmacologist. Those are the people who try to understand what they call ADME, absorption, distribution, metabolism, and excretion, meaning understand the drug when you get in the body, how the drug is metabolized, absorbed, and then secretion. They're trying to understand different people and the different conditions, how that can affect the drug. For example, elderly patient, young patient, kids, pediatric, people with the liver disease, people with the kidney disease, and their drug-drug interaction. So there are also rules for physicians like me. In clinical studies, we are going to deal with clinical patients while we are not treating patient, but we give advice to the treating physician about the drug and work with the physicians, normally principal investigator, that's something we can understand their language and try to deal with this. There's two aspects of it. One is efficacy. So as a physician, right, you probably understand more about the disease compared to treating physician. We do a lot of literature review and try to understand the scope of this field. As a physicians, their mind is to the best of an individual patient's benefit. For us, is drug and patient. And the other great aspect is safety. Safety is very important, especially for those chronic diseases, KX826, for example, the chronic diseases. The safety is very important. The limitation is that in the clinical studies, there's only a limited patient. Then in the post-marketing, that's something, you can have big patient exposure. Patients have different kind of underlying disease we're using. You'll understand more about how this drug works and the different conditions. So that's something that physician's job. Along with that, there's a lot. I don't want people to get the impression you have to be a physician to be involved. I saw a lot of people. I would say, Kyle, I think if you want, you can probably get a job in the pharmaceutical industry with your knowledge. But there's other people who can also involve, like for example, like even you have an undergraduate degree. You said, as Kyle mentioned, bioinformatics. Those are very hot areas that involving AI, that involve a lot of things, because the information is the key. Used to be, we are doing things in silo. Say even for me, like I do things for the company, I know something. But you know how many companies there is? Well, a lot of companies. Do I have enough time to look for the literature? No. So those are the things, information is the key. So if you put it like now we have AI assistant drug development. If you put enough information, train the computer enough, you can predict a lot of things, not all of them, but a lot of things. Those are the career. I see people with, I can tell you when I'm working a big pharma, people with the PhD in zoology become the lead clinical development. What's needed is very good logic, very good stats, and very good logic to lead. So that's that. There are also assistant. For example, we have to have people that post-market collect the information, collect safety information, and put it in the database, and summarize and write a report to the FDA, to different regulatory agencies. So people with a good medical writing skills are also needed. There are also different roles. One thing I forgot to mention, stats. As you probably know, that we're also talking about possibilities. Statistical analysis is a key to this clinical study. For example, how many patients is needed, right? So how it depends on how many, how big the sample size depends on how big the effect size. If you have a drug very efficacious compared to placebo, then you don't have to have a lot of patients. If they have a drug that's marginally better than the drug, the placebo, then you probably need a sample size. But people don't think that's not a good thing, because sometimes you have a drug, especially you're talking about head-to-head comparison, right? So then you talk about the more sample that's needed. So those are the things, and also there's a data manager. I know there's a lot of people in college and study the data management. That's also important. Everything we said, well, in the, as I said, patient reported outcome, and also whatever the primary endpoint, or whatever the parameter, those are data. Data will be entered in the database, clinical database, how they enter, what's the formulation, right? You can imagine, as Carl said, multi-center, sometimes global multi-center. your path, your way to enter is not the same as different as other country, right? You even the month, date, and year, and for other people it's date, month, and year. Those are things need to be controlled. So a good data manager will identify all those issues, make a good study report, and then case report form, and so that we can capture. So in a sense, almost everything to life sciences, you can find a way in the pharmaceutical industry. But as I said, you're gonna have to have you have to have a persistent. You have to work hard. You're gonna have to, most importantly, deal with the setbacks. And the other thing, the final thing I want to mention is that whether we are, I'm not saying we, in general speaking, we are special is because if you look at the industry per se, sometimes we're, and hardly any day go by you didn't hear anything say, oh, this company is laying off people. That says that we're hard, or it's a hard job, because sometimes you work hard, you think you get a good product. Sometimes for various reasons, the drug didn't show that. A lot of times because of our company's existence depends on investment. Sometimes it doesn't work. Sometimes it will lead to lots of layoff. Sometimes even for bigger pharma, if the pipeline was not catch up, so sometimes it also have a lot of effect on the stock price, which lead to the layoff. The recent Pfizer is a good example. I think in the COVID area, everybody knows Pfizer, but if you look at the current price, the stock price is half because they didn't have the follow-up drug out quickly. So another way to say it, Colin, sorry to take a little longer. I'm just giving this a long-winded answer. I also
No, it's fine. Yeah.
ask me for some advice. So I just take this opportunity. My apologies.
No, it's fine. That the advice is well received. Yeah. Grace, do you have anything to add? Maybe any sort of advice of your own for anyone trying to get into it?
Yeah, Wilson has already, with his Tom's experience, he already gave a very good guidance for anyone who would be interested in this field. I would say, pharmaceutical, especially when you mentioned pharmaceutical, it's only part of the whole, the only part of the process to able to provide the effective medicine or device to ultimately reach the people, the patients who really need it. And upstream and downstream, upstream there is preclinical fundamental research, and also downstream there is market and real world, a lot of data like Wilson just mentioned. So I would say, if you're interested in the field from all the perspective, drug development itself, or from data perspective, or from, just the research perspective, and from regardless what reason, like yourself, if you are interested, or from your personal interest, or even you are cross field from the other career, if you are just interested, I think current advanced technology, especially with the large exposed to different social medias channels and chat GPTs, you have a lot of channels to just starting to take a look and do some sort of not real but just from knowledge absorption. You have a lot of resource just to try to understand what's exactly going, and try to connect to what you are current stage, regardless if you're a student, you're in a college, or you are from different career. As long as you're interested, there's a lot of channels you can speculate those knowledge and find a way and find connections from your current situations. If you're a student, you can pursue like biology, or or chemistry, or data science. All those career can eventually lead you to the pharmaceutical, and of course the drugs and effective drugs and devices. The ultimate goal is to save people, to bring more effective ways to those people who really need this huge impact. Just from that perspective, that's a very meaningful job. And I would just say, if you're interested and you're able to, and prepare from education and try to explore as much as you can before you really get into the field. And even in your field, for example, myself, I have been navigating in different positions and better understanding, have a big picture of how those big drug development and how it's eventually reach the patients. So that's very fun journey and very interesting. So from that career, I think the modern life, the current well development artificial intelligence, ChatGPT, and all the channels can bring you very unique time and very unique opportunities for anyone who is interested. There's a lot of opportunities and possibilities if you'd like to divide in. Yeah, that's my in general advices for anyone interested in this field.
I just want to say again, thank you to both of you guys for coming on. And I really appreciate it. This is an awesome interview. And I think KX826, for those of you guys who don't know, that is KX826. You guys, I think there's a holiday sale going on, maybe because we're right around. Yeah,
absolutely.
Yeah.
Yeah, so we're working on the poster should come in soon. Either starting from December 20 or 21st, and then I need to check with the team. But Cal, you should be able to see those posters be listed in the Koshan Mall. And in additional, just for your personal, anyone, any audience who subscribe to Kyle or who is interested or watch the video by chance, there is additional, discount through the link, your personal link. And just to give the chance for anyone who would like to try on the products. And there is very good price discount right now. Yeah, just go ahead to check.
Thank you so much. Well, that's the end of this interview. And yeah, have a nice day, guys. Really appreciate it. Bye.
Thank you so much, Cal. Yeah.
Thank you.